The overall goal of the following experiment is to identify rare epitope-specific T cells from the endogenous T cell repertoire of mice. This is achieved by first staining harvested lymphoid cells with peptide MHC Tetramer reagents, which will fluorescently label the relevant epitope-specific T cells. These tetramer-labeled cells are then stained with magnetic microbeads conjugated to antibodies specific for the fluorescent tag on the tetramer. Next, the magnetically labeled cells are passed through a magnetized column in order to enrich the sample for tetramer-bound epitope-specific T cells. Finally, the cells are stained for the desired experimental cell markers, and multi-parameter flow cytometry is performed to detect and quantify the rare, tetramer-enriched epitope-specific T cell populations.
The main advantage of using this technique over other existing methods, like the TCR transgenic T-cell adoptive transfer system, is that studies are performed on real polyclonal populations of epitope-specific T cells that develop naturally within the immune system. Moreover, the high sensitivity of detection afforded by this technique enables studies of these populations when they are present at extremely low frequencies. This method can help answer many key questions in the field of immunology, such as how many naive T cells may recognize a given antigen from a pathogenic microbe, and how the clonal composition of an epitope-specific T cell population changes throughout the course of an immune response.
Before starting the dissection, add 1 milliliter of complete EHAA medium.